Uh, Francesca, are you ready?
Yes, do you see my presentation?
Yes,
Okay, so
everything is good. So please, you can start 30 minutes and leave couple minutes for questions.
yes, please interrupt me at 22, 23 so that I leave space. Thank you very much for organizing this excellent workshop and for giving us more time to speak about our subject so that we can start maybe also a new collaboration or strengthen the one that already exists. Sorry if I will not look into the screen. I look at the big screen in front of me. So uh, in this talk, I'm speaking about part of my research that started 25 years ago on nucleic acid quadruplexes, and I will speak about the latest results on the stabilization of the structure with ligands. I'm coming from the University of Cagliari in Sardinia, Italy. And I have a long-standing collaboration from almost 10 years now with Dr. Peripilizia from the National Academy of Science in Ukraine, and I am very, very happy to be part of this meeting. Uh, so I am aware that the quadruplex structure are not known from most of the public, and I expect that also in an audience of theoretical physicists is important to introduce the subject and why it is important to study the ligands that are interacting with this structure. And then I will show a single example of study that we performed on this system. And I added some more information on other study that we have performed and on some study that are currently performing and for which maybe we can start some collaboration with experimentalists. So here is represented one chromosome and the structure of the double helix, and also some structure that is not a double helix, but it's actually a quadruple helix. Indeed, it is known that since basically the same period when the double helix was discovered, it was known, it was shown that if you put guanine in a solution, we form a gel whose structure is due to the formation of quartet. Of course, this depends on the type of iron and the concentration. A quartet in which the base pair do connect not only to a single base pair, like in the Watson-Crick base pair, but base pairing that we observe in the duplex, but they can form hydrogen bond with two bases at the same time and form structure of this type. Also, triple helix exist. And in, in at the end of the last century, it was known that there are in the chromosome of all the living species sequence that are particularly rich in guanine that can form more easily this type of structure compared to adenine. And it was known that if you cut this sequence and you work in a certain condition in solution with ions like potassium, but also ammonium and other, they can form quadruplexes structure. And so since we see, and in the end of the chromosome of the living organism, there are repeating sequence rich in guanine that can form quadruplex. So if a structure can form, it's probably that it has a function. So this was thought since long time that they might have a biological function, but only in this century it was demonstrated through several techniques from several experimental groups that during a particular stage of the cell, here is shown the metaphase chromosome of cervical cancer cell with some regions that are highlighted by fluorescence, and this fluorescence is observed because a binder specific for G quadruplex becomes have light, give the fluorescence phenomenon when it is bound to a quadruplex. So this is considered the demonstration of the existence of the quadruplex actually in the cell. For many years, when we were studying quadruplex, it was yet not completely sure that they existed. There were a lot of indication, but not really proof. Now it is accepted by the community is a fact that the quadruplex indeed form in the cell, not only in the lab when using small sequence. And the bioinformatics studies have once we were able to sequence all the base that are part of our genomes and the genomes of all the living species, of many living species. This gave space to study of bioinformatics in which the sequence can be analyzed by computer and check if there are sequences where the guanine are in such an organization that can lead to the formation of quadruplex. And limiting our discussion at the moment to the human genomes, there are really a huge amount of portion in our chromosome where the sequence can form quadruplex. And what is interesting is that this region that can form quadruplex, that are called putative quadruplex forming sequence, PQS, are not located randomly along the DNA sequence, but they are more frequent in very interesting region of the chromosome. In particular, what attracted initially the interest was their presence in the terminal region of the chromosome, but they are also are overrepresented in the promoters region of oncogen. So oncogen are genes that when they are activated or overexpressed contribute to uncontrolled cell growth and therefore to cancer. And also the telomeric region are involved with cancer because there are some protein telomerase that keep this sequence at the end of the chromosome longer and longer. This sequence at the end of the chromosome have the role to protect the chromosome. So when this sequence during the division gets shorter and shorter, this leads eventually to the cell death. But if the protein of telomerase prolong this region, the cell get basically never die. And this is very bad because from the cancer point of view, because this protein, the telomerase that has this action, is particularly active when in cancer cell. And what is the role of quadruplex in this context? The role of quadruplex is that if the structure reaching guanine form a quadruplex structure, then the action of the proteins that elongate the chromosome or that start the reading of the genes that are connected to cancer, if the quadruplex are formed there, then this protein cannot act and the cell dies. This is the reason why the control of the formation of G quadruplex has attracted a lot of interest from the scientific community. And in particular, there has been a lot of attention in finding molecules that can stabilize the quadruplex, because if the quadruplex is stabilized, this can help us in the find of cancer, but not only cancer. In the last 10 years, quadruplex have been considered as a promising target also in the fight of viruses. So here the quartet structure is schematized in this way to show how a chain of DNA can fold, forming a quadruplex structure in which are present two or more quartets. As you can see, there are multiple ways in which a single chain can fold to form a quadruplex. And this is very different from the structure of the double helix of DNA, where there is only one way, two chains that are running antiparallel. While in the quadruplex, we can have the fragment of chain having the same direction or opposite direction, and many topologies. Here are shown 4 topologies, but there are many more topologies. And you can see that they differ, for example, in the exposition of the quartet to external agent, as an example. A common features that the ligand that interact with quadruplex should have is the presence of a large planar aromatic surface, because such features allow for the stacking to the external tetrads. And in the study, in the example that I'm showing in this talk, I discussed a study that started with a colleague from the National Research Center of Italy, that is the analog of the National Academy of Science in Ukraine, proposed to us the study of a class of molecule, PBTs, that have this core and different residues attached in this position that I'm indicating with the mouse. And he had synthesized this molecule, and he had already checked that this molecule, whether this molecule had some anti-proliferative activity for tumor cells. And his work had already shown that they were active against about 60 tumor cell lines. However, his study could not demonstrate whether the interaction was duplex DNA or quadruplex DNA, and asked us to check whether the role, their activity, was due to the interaction and stabilization of quadruplex structures. So, in order to answer to this question, we used as a model of quadruplex structure of relevance the quadruplex structure formed in the promoter region of the oncogen CMYC. This that has this sequence TGAG as reported here. And we first performed a series of docking studies on this structure using for the docking the Schrödinger package suite. And then on the most promising compound and on the worst compound, we perform molecular dynamics simulation and also experimental study through a colleague still of the National Research Center of Naples, Giovanni Roviello. We perform a CD study to verify experimentally the stabilizing capability of the ligands. So, focusing on the docking results, we studied, sorry, I go back 1 second. We checked all these compounds that differ in the residues attached in these positions, 8 and 9. So we had these residues. So we prepared the molecule in the software Schrödinger, and we calculated the different docking poses, which means different way of the ligand to bind to the quadruplex, and the energy according to some energy formula that is characteristic of the program and is known as G-score. And here is reported the energy in kilocalorie mole. The value on the top are higher values, so less favorable interaction, and the value on the bottom are lower value, which indicate more better interaction. And we use this different method, either based on classic for field, either based on quantum mechanics, and also different way of calculating the energy. And what we can conclude from this analysis is that one compound seems to be better than the other with different scoring function, and one compound seems to be worse than the other. For the other compound, we have intermediate values. And we wonder, when we perform docking, we have a value of energies, but what does this number mean? What does a value of 5 really indicate? Does it mean that there is a good interaction or not? In order to check whether these values are corresponding to this compound are good or bad, what we do is to take the database of molecules that have drug-like features, which means that they are soluble in water, they can form hydrogen bond in a certain number, and so on. And we took a compound with a similar feature to ours, in that similar size and absence of charges. And we checked, does our class of compound perform better compared to random compound or not? And we were hoping that they would perform better. But actually, if we consider the value here of our compound, they are highlighted by this colored rectangular shape. And there are many compounds that have a lower energy for any of the score that we considered. So according to the docking, these compounds are not so greatly promising for their interaction with the quadruplex. But nonetheless, we performed more accurate work using molecular dynamic simulation because the docking is performed on static structure while these molecules are highly flexible. So the binding to static structure, while the structure can change a lot, has a lot of limitation. One of the main limitations is also that in the docking, we don't consider the role of the solvent, just to cite one. So the docking give a quick result, but these results have many limitations. Molecular dynamic simulation have less limitation. So what do we do with molecular dynamic simulation? We simulated our system, our quadruplex, in presence and in the absence of the ligands that have the best score in the docking. This one on the right is the compound that has the best scoring with these two substituents, and the worst compound is the one that have the that is here in the center. Here are the results for the quadruplex alone. And what are these results that I'm showing? These results are root mean square deviation. Root mean square deviation are values of a number that indicate how much a structure differs from its initial or experimental structure. So if our quadruplex is denaturing, the root mean square deviation will rise very much. If the compound is not denaturing, the root mean square deviation will be stable. We simulate the compound at a temperature near that of denaturation of the compound alone, but we cannot expect to see the denaturation at the same temperature that we observe at which we observe denaturation in the experiment, because the denaturation occurs in time scale much larger than 1000 nanoseconds, that is the length of the simulation that we can perform. So in order to speed up the denaturation, we performed simulation at higher temperature, 500 Kelvin, 550, 600 Kelvin. Here, high temperature simulation are not intended to reproduce physiological condition at that temperature, but are rather used to provide a comparative framework between system modeled with the same force field, but different ligand or absent of ligand, and assuming that any thermal artifact that occurs at 500, 550, and 600 will be affecting all the system that we want to compare in the same way. And what do we observe for the quadruplex alone at 500 Kelvin? And for the quadruplex with the worst ligand and for the best ligand, we observe that there is no denaturation. If we increase the temperature to 550 Kelvin, we see that with the quadruplex without ligand and with the worst ligand, we observe denaturation at 550 Kelvin, while at this temperature, we do not observe denaturation for the system with the best ligand. What does this mean? This means that at the same temperature at which these systems are not stable, if we add the ligand, they are stabilized. The idea is that higher is the temperature at which the system shows denaturation, higher is the stability of the system. When we perform the simulation at 600 Kelvin at even higher temperature, we can see that in presence of the worst ligand, we have denaturation at in the early stage of the simulation, while with the stabilizing ligand, we have denaturation after a longer period. Here are shown some snapshots from the simulation that show how the best ligand move from the external tetrads during the simulation. And when bound to the tetrad, stabilize the system, while in the other case, it left the quadruplex before. So now let's consider the system with the experimental results. The experimental results agree nicely with the simulation because the highest melting temperature with circular dichroism is observed with the best compound with the docking that is observed to stabilize the system also in the MD simulation. And we can observe that the melting temperature with
Minutes, please.
Yes, is higher than the simulation of the quadruplex alone, while on the opposite, with the worst ligand, the melting temperature is lower, or if we consider the error, similar to that of the quadruplex alone. And this also nicely agree with the inhibition anti-proliferative activity expressed as EC50, which are the lowest and therefore the better for the best compound that we have studied. So to conclude, the G quadruplex are stabilized by this ligand that show anti-proliferative activity against the experimental against the tumor cell, and so therefore the possible mechanism of biological activity this ligand is due to the interaction with the G quadruplex. The simple docking is not able for this ligand to suggest a particular capability of stabilization, and we propose that to verify the stabilizing capability of the ligand using simulation at high temperature. I want to thank you very much for your attention.
Thank you, Francesca. You are in time, and we have time enough for questions. So please, questions in the room, questions I can see Andrei in Zoom. Please, Andrei, your question.
Thank you. Thank you, Francesca, for a very interesting talk. I was wondering if there are any other ions except magnesium that can stabilize quadruplexes or maybe some other oligomers?
Could you please repeat the question? I didn't hear very well.
You showed that the magnesium ions stabilizing quadruplexes. Are there any other ions that can do the same?
So all the ions like sodium and potassium stabilize the quadruplex. Lithium not so much, and also ammonium can stabilize the quadruplex, and also other monovalent ions. So basically thank you very much for your question. It's a very important question for the quadruplex because the presence of the ion is a key point for the existence, and the nature of the ion is a key point for the existence of the quadruplex and also for their particular topology. So, for example, with some ions, a certain sequence can have stabilized this type of topology, but with another ions, another topology might be more stable. So they are important not only for stabilizing, but they can stabilize different sequences. And for us, from the molecular dynamics point of view, is an headache.
I see. Thank you.
Okay, thank you. There is one more question in Zoom. Alena Dobeshko, please.
Yes, thank you, Francesca. Quadruplex is very interesting topic, and my question is about it is proper slide. Now, my question about these different forms which presented on this slide. I want to ask, did you study this form using vibrational spectroscopy, infrared or Raman, for example? Because maybe it's more sensitive than CD method, which you showed us.
Thank you very much. This is a very, very interesting question, very important point. So far, there are no, so while, for example, in the protein, we have difference, for example, in the carbonyl vibration, depending on the secondary structure, and there can be a little variation that can indicate, can be deconvolved. Not such study has been performed on the quadruplex. A main problem is that the peaks are very, very close because there are both in the infrared, but also in the NMR, which is, NMR is used a lot, but infrared not so much, because I think the main problem is that it's all due to guanine. The quartet here are all guanine, so very, very similar. So I think it's very difficult to deconvolve because there are so many similar peaks. So infrared, there is no single study where I saw infrared used to study the topology through infrared. So to know the topology in a quick way, only, well, circular dichroism, but also FRET are used. And if you have a lot of time and power and very good people to work at, NMR spectroscopy, not infrared.
But it is necessary to probe, to try.
Yeah, I think it's a very interesting point that you're raising. For some reason, I never checked particularly for that. And I will check what are the limitations. It's a very, very interesting point.
Okay.
Thank you.
Thank you.
Three. Okay. So more questions? Maybe I have a question. Francesca, maybe you can comment this. How these structures are, these quadruplexes are stable if one changes pH in water environment? Does it affect, does pH affects the structures? Maybe somebody studied this.
Okay, so let's say the protonation state of the DNA itself, unless you go to very lower and higher value, does not change much. I am, so in general, the study that we perform, we try to be near the physiological condition that are of a pH 7, pH 7.4. And also the interaction with the ligand, this can be somehow very much affected by the pH. So we take into account also the protonation form of the ligand, typically at pH 7.4. In general, we don't vary for this application in the biological, with biological pharmaceutical scope, we don't vary the pH. But the point you're raising is very interesting for technological application, because quadruplex can be used also, and are used also for technological application, because the ion goes in the center of the channel, even if it's not represented here. you can have, by stacking many quadruplex, a long chain of ions that has been used. But still, and I think there it's very relevant, the pH, but still I cannot really I don't really know how it varies, the structure. I'm not aware of the study for the variation of the pH, but I think it can affect. It can affect because it will affect the solvation, and the solvation is important for the structure.
Okay, I see. Thank you, Francesca. Is there more question? Okay, if no questions, we can send Francesca again.
Thank you.
And we have next speaker.